Just trying to get. Okay, we got our recording in progress. Sorry for the brief delay there. Um, I am Chuck Sywin. I'm the Director of Small Molecule Neurotherapeutics Development here at NINDS. And I'm happy to tell you about the Blueprint Neurotherapeutics Network today. I guess I should probably turn my camera on. Let's see if I can do that. I don't usually, but let's give it a whirl. One moment. Morning. Oh, I guess it does not let me do that. Never mind. Thing. Nope, definitely doesn't. Okay, very good. We'll move on. So thanks for taking the opportunity to learn more about the Blueprint Neurotherapeutics Network. The BPN, as it's known, is housed within the Division of Translational Research at NINDS. And the staff who supports the program, this Trans-NIH program, is all pretty much at NINDS with some assistance from NIMH as well. We have as a goal to, in the Division of Translational Research, to accelerate basic research finding towards patients. And we do that by having a suite of funding opportunities, either through grants or access to contracts, access to consultants and training across the spectrum, from assay development to sort of that first in human early clinical trial development. And we also do it for different modalities, for biologics, for small molecules, for devices. I'm going to focus today on the small molecules, but I will touch on the early stage program to get you the preliminary data at the end, as well as mention that we have biologics program set up is very similarly. The Blueprint Neurotherapeutics Network is part of a suite of programs. As I mentioned, it has two different tracks. It has the Small Business SBIR Cooperative Agreement Fast Track. This is under PAR 20-111, and it is paralleled by the BPN Biologics program as well. So everything you hear me talk about today is also available for biologics. There's some small differences, which I'll touch on when I go over the BPN biologics programs at the end. The UG3 UH3 is PAR 20-122. This program is for all comers. And it can have foreign components in it. It can have even foreign institutions in there. They both have the same receipt date, August 9th, coming up. And just know that this SBIR is for SBIR eligible institutions. And so you have to self proclaim that you actually are eligible for that program in order to be applied under this. Both programs have clinical trial optional. And I'll talk about it. What that really means is whether you're doing the clinical trial yourself versus our contracts. In all cases, the program is designed to take you all the way to the clinic. We do not focus on assay development or target validation work. However, that doesn't mean that the assays can't be optimized as part of the early stages, from where you start to a more appropriate level for the throughput required. The vision of the program is quite simple. We combine the strengths of NIH and industry expertise along with small molecule neuroscience drug discovery and development to move your projects forward. And we do this by you coming up with an idea on novel drug targets or strong, hopefully with strong disease models and assays in place to execute the program. We can provide advisors with extensive pharma experience to help you move those programs forward. We have a suite of CROs already in place to fill in the gaps in your investigator team to move the projects forward. With with the combined goal of de-risking the programs and advancing to clinical testing and hopefully hand off to licensors that will have deeper pockets to take them on into the phase 2, 3 stages as things go on. There is a program goal, and I'll touch on it many times throughout the thing, is to maintain the IP and intellectual property to your granting institution, and we do that through a number of different means. Bottom line, de-risk the project. We always want to get to investment. We want to see you take the projects further. We want to see this get to patients. We are looking for the best ideas, and we're looking to fund those through this mix of infrastructure and funding. What's really good about the program is it's non-dilutive funding, and it provides the necessary resources that are typically lacking in many smaller companies as well as academic research facilities. With the goal, as I said, to facilitate licensing, we try to preserve your intellectual property to move things forward. It's open to academics and small businesses. We move things to the clinic. They can enter, any project can enter along the paradigm from early drug discovery to development stage. Very few things are actually truly development stage projects, but the way we define development stage in this particular case is things that are, you picked your compound, no more just medicinal chemistry is required. You will typically be considered at a development stage for at least the first pass of discussion on what data you have and what you don't have already. And then discovery phase are things that are certainly optimizable. Reason why I say many projects don't actually make it to are not really development stages, they lack a lot of the data package that's required to be truly a development compound that you've de-risked appropriately for entering sort of the advanced clinical or preclinical studies like GLP tox. The opportunity to get access to our different consultants and contracts and the science officers and things comes with the program, but you do have to lay it out in your application. And part of the purpose of the application process is tell us about your project, but also tell us what you need for resources and make sure that we are a good fit for what you're doing. And to that end, we always ask you to give us a call before you apply as a best practice. It'll make your application process easier. It'll also ensure that your budget is accurately reflecting what we can and can't do to help you out. So what can we do to help you out? The infrastructure is set up so that we meet in a lead development team every 2 weeks, just like you would in pharma. We have a cadre of consultants across the drug discovery and development expertise that are required to move a project forward. We have NIH staff, expert program managers with drug discovery and development experience to help facilitate the project teams. We have science officers that really understand where your project fits into the overall portfolio of projects NIH is funding in this area of research and can make sure that we're doing stuff that is truly industry practice, truly at the cutting edge of what is known in the literature out there for the most relevant models and testing that we could be doing. As I mentioned, you get NIH dollars for supporting yourselves at a minimum you have to have the bioactivity and the efficacy studies. The disease biology portion has to be in the grant side for sure. Because we have hundreds of diseases, we can't provide that across the board to you all. But we can certainly augment it with knowledge from staff, and you can have co-investigators to do this kind of work. We have a suite of contracts that handles everything from medicinal chemistry, and medicinal chemistry is not just synthesis. It's things like in vitro ADME, it's things like computational chemistry, structure-aided drug design. It's our PK tox contract can do both rodent and large animal PK. It can do non-GLP dose range finding and GLP toxicology for IND enabling work. My recommendation is, we can certainly handle the large animal PK. If you have money in your budget and room in your budget to include the rodent PK, you can do that reasonably well. in many cases. If not, we can certainly do it through our contracts. Um, I'll skip the data management piece for a second. We have drug manufacturing formulation. This does things like demonstration batch, scale up for the tox, scale up for the clinical trials under GMP. It does analytical work. It does formulation work like dose screening, salt screening and polymorph work. It does things like figuring out what we're going to do for the clinical side. We can do API in a capsule. We have to blend the capsule to the solution. How are we going to do the clinical trial for phase 1? And we have clinical trial contracts, primarily focused on doing healthy human subjects. And if you need to do something beyond healthy human subjects, you should probably have a conversation with us to make sure that whether we have the capability to do that or whether it's something you're going to have to do yourself. So always think about this as, do you have the expertise to do it in-house? If not, talk to us and make sure we can provide it for you within our contracts. I skipped the data management piece. This is really designed as a relational database. If you're optimizing your molecules or as a central repository for your data, it can also be a great way to get it back out later for when you're doing licensing conversations. If you keep everything in one place, it's easier for people to export it out for things. As I mentioned, it's tailored to you, it's non-dilutive, and as I said, we protect intellectual property in these contracts. The medicinal chemistry, drug manufacturing, formulation contracts, for instance, have determinations of exceptional circumstance clauses that are contractually bind these contractors to assign intellectual property to your institution for the work is being done. And that protects your IP and make sure that you can move to licensing easily. As I said, this is a trans NIH program, and there are many ICs involved in it. This is the list of the ICs here, and the individual science officer contacts that are in here are available to discuss your program. Keep in mind, being a trans NIH program, each of these ICs is paying for the particular project in their mission. So if it's drug abuse or opioid use disorder project, for instance, NIDA will be paying for it. If it's a Alzheimer's project, in most cases, NIA is paying for it, etc. So you just have to know where it is, and we'll talk with those. But you can always start with have a conversation with me, and if we can talk about what the best indications. I know in many cases people say, I have more than one indication, which one should I go for? The answer to that is quite simple. Go for where you have the best set of data or preliminary data to support your hypothesis. And that will be probably the place to get the funding first. Down the road, expansion of indications is a normal part of drug discovery and development and will probably be whoever you license it to will work on that. The exit point, as I've talked about licensing a number of times in here, just like the entry point is along the pathway, the exit point's along the pathway. We've had people exit as early as lead optimization and as late as the end of phase 1 clinical trials with us and everywhere in between. So again, we are going to, as your project progresses, you may or may not be in a position to license it earlier or later, depending on the indication area and the data you've generated to date. Being a two-phase grant mechanism, again, I get a lot of questions about this. Everyone must enter the preparatory phase. In the UG3, this is a 1-year phase, and it is really designed to sort of kick the tires to make sure that what you say in your grant is executable. If it's an early stage project where you resynthesize molecule and it still has activity, the assays have the throughput that we're talking about and are performing the way we expect it to drive an SAR program. If you're coming in at a quote development project, this preparatory phase may actually be doing things like dose range finding tox, or it might be getting the in vitro data package required for transporters and DDI stuff that the FDA now requests for most projects coming in. So we'll look to that and determine what you need. And again, a preliminary call with us is probably a good idea. to determine where you come and what you should do. Now, I said UG3 is a 1 year. It is possible, because phase 2 of a U44 is limited to 3 years. If you need 5 years, it is possible to come in for a 2-year phase 1 award under the U44. I say it's possible, but it's often difficult to do because you are limited by the SBIR rules of 700,000 in total costs if you have invertebrate animals in phase 1. It's even lower if you don't have that. So that could limit your ability to stay in phase 1 for 2 years. And so oftentimes, if you're in a later stage project in U44, we have to consider how we're going to do this. If you need the full 5 years in U44, again, contact us. I'll be doing more through the contract side, and we'll work through how to make this make sense for you. Each project can only receive one UG3. So you can't come to the end and say, well, we can't do this, restart UG3 again. It's all milestone driven. We make decisions on transitioning from the preparatory to the execution phase for every project. And not all projects will proceed, because some projects just, when we actually kick the tires, things don't work out. We say really need to go back and get additional data. Additionally, as you go through the projects, we can have, and we do have, annual go-no-go decisions on each project moving forward. And you can, in fact, stop anywhere along this path for scientific reasons. We hit a wall, we can't get there. It could be for competitive reasons. You're just scooped out, you no longer have intellectual property, someone else is in your area, and you really have to retool and go back. There's multiple reasons why you could potentially stop midstream, but our goal in all cases is to move your project to the clinic. That's what we want to see, and get there. The other part is we can do limited medicinal chemistry in this preparatory phase. So you can get up to 3 years of support, but it's at the first year's at a lower level of support, as we really are, as said, redoing preliminary SAR, redoing, if you come to the exploratory phase, the assays, trying to show things are getting tested, benchmarking those compounds and so forth to determine what's going on. If you're coming in at a later stage medicinal chemistry, there'll probably be a discussion around, do you have it properly benchmarked or not? If not, we may call it exploratory, because we need to do that benchmarking first before we go into hit to lead. Again, my advice is, do not try to put yourself farther down the path than you really are, because that just makes getting milestones more difficult. I'd go past the slide. I did not. Okay, sorry. This is sort of a recap, that preparatory phase. It has a note here to remind you, or remind me to remind you, that not all institutes or centers accept development projects. And so, for instance, NIDA and NIMH specifically have language saying they won't take a development project. In NIDA's case, they have other funding mechanisms set up for doing that, that they prefer you to go through. I mentioned these costs briefly in the 700,000. These are sort of the guidelines, but in all cases, you need to talk with us and make sure that you are properly funding what you do. I do not recommend trying to under budget yourself in there in order to meet the guidelines. I would rather you have a conversation with us about what we can do through contracts, what you can do yourself, and determine what the best path forward is for your project team. I did mention that the UG3, UH3 mechanism, the 20-122, can take foreign components. It also can take foreign organizations, and we do get applications from foreign organizations, and we have in fact funded applications from foreign organizations in the past. But the reviewers will assess whether the project presents special opportunities for further research programs through the use of unusual talents, resources, populations, or environmental conditions that exist in those countries but not are readily available in the U.S. or augment existing U.S. resources. And the case has to be made by you, the applicant, for why this is. It'll be reviewed by the review panel, but also before award, it has to be reviewed by council and approved by council for any foreign award. The entry criteria, as I mentioned earlier, you have to have the disease biology in your shop. You have to have the assays to run a lead optimization work in your shop for the disease biology, at least the primary and selectivity assay, the counter screen, and so forth. You must have a starting point in terms of chemical matter. That starting point can be a literature compound. It has to be amenable to chemistry, and the IP has to be free of obvious roadblocks. So if you're starting from a literature compound, um, then you need to be thinking about how am I going to scaffold hop to a new area that's going to be, an area I can get intellectual property. Some of the common pitfalls I see for people is they come in and say, I want to take, Pfizer's compound X and put it into a clinical trial. I'm just going to do it. And it's like, well, you don't own the intellectual property on it. You don't have an agreement with Pfizer to do this. How are you going to progress this, and how are you going to have something licensable at the end that someone's going to invest the money to do phase 2 and 3? So think about it in those terms. Who's going to pay for those big clinical trials down the road? Do we have, is there a path to get there? Because that's the same lens that we'll be looking through at the end of the day. How do we get this to patients? Is there a path for us to get it to patients? The development is a fully optimized compound. No more medchem. You have to have strong data package in there. You should have the biology and admin for the intended clinical use. You should have an efficacy in there done. Ideally, you'd like to have at least a dose ranging finding in rodents so you have an idea of the therapeutic index. You have to have a feasible path to the clinic, and that feasible path means I have to have intellectual property. I have to have synthesis ability here. So if I have a 17-step synthesis that can only make a gram of material at any kilos, that probably is not going to fly in terms of work. So we'll have to figure out, that's going to be the biggest hurdle. So just be aware what your hurdles are, and move the project forward. In many cases, that's going to be part of the work that has to get done. I'm sorry, this has popped up here. I didn't realize that. The last step I want to talk about is just these milestones. We have it pretty well mapped out for small molecules, what we need to do as we go through the different stages. As a minimum set of milestones, they follow the drug discovery and development pathway from early stage thing where you have, confirm you have the assays, the screening funnel, the target product profile, and you benchmark your compound, all the way to, as I mentioned, development world. You have the compound, you have a tractable synthesis, you have a stable salt form of polymorph figured out, you've got large animal DRFs done, you have your clinical specialists done, and you're moving into IND enabling and eventually into humans. We'll work through this. We assign these milestones to your program based on what you put in, but it's a good way to lay out your program timeline as well to follow this within that 5-year time frame to get from where you start to the clinic, how long it should take you to get there, and what we can do for contract support. I'll touch on this one more time because this just comes up over and over again for me. Intellectual property exploitation. Inventorship is inventorship is inventorship. That's dictated by law. There's no way around that. So what we do, our consultants and our contractors that are going to be doing synthesis and stuff for you, they will be doing inventive work. They're going to sign IP agreements, and the consultants, the contracts have clauses in them to make sure they have to sign IP agreement to your institution of record on the grant. NIH staff are here to facilitate, not to tell you compounds to make. So we facilitate things, we're on the meetings, but we're very careful not to be in the weeds to the point of trying to become inventive on our side. So we're trying to seek no stake. We're not trying to actively get stake for us. We're trying to actually do the opposite. We're trying to be facilitators and make sure that we're not inventors. Prior to a grant award, the PIA institution must have upfront IP agreements with all your co-investigators who come from different institutions and likewise. And I'll be asking you for that, especially from multi-PI grants and things. I want to know who it's all set up, which institution is going to be the lead. They're not going to have any problems filing the intellectual property when we get there. If you have royalty agreements, that they're put in place in advance, if any are required. The IP should in all cases aim for unencumbered IP that's consistent with our program to create a licensable project at the end. We have a website. Here's the link for our website. And on this website is a treasure trove of information. I point you to the left-hand side. It has the funding opportunities, has our current portfolio of project. It talks about the different models we work on, who's on our external oversight committee that helps us evaluate your milestone progress. Talks about the resources, the individual contractors and consultants that are available on the thing. You do not pick the consultants and contracts. That's based on availability and sometimes competition for contractors. And we have a whole frequently asked question section here. The same contacts that I showed you earlier are available. And just as a sort of a parting thing for the program, I want to show you, the fact that the program does work. We've had at least 10 projects have announced industrial funding since starting the BPN. We have a number of projects in the clinic that have announced clinical work since either being in BPN or since leaving BPN and continuing. We have 2 projects that publicly have said they're in phase 3 presently, and other projects that are in different phases of clinical investigation. So I should mention that we have these early programs I touched on earlier. Things like Ignite can help you set up your assays, develop animal models, do in vivo efficacy testing. And the Ignite program is innovation grants to nurture initial translational efforts. There are 3 different PARs for each one of these different areas. Develop an assay, run a screen, do some early med chem characterization of your molecules and run in vivo efficacy studies to help you get that preliminary data that will shore up a BPN application. Or if you need to develop an animal model system to facilitate the work, there's funding opportunity. These are phased R grants. They're milestone. They have to have a milestone in them that go from one phase to the other. They're up to $499 in direct costs per year, up to $750 in direct costs for the total project across 3 years. It has application due dates every 3 months, or sorry, every 4 months, 3 times a year. And it's extremely important that you have clear and quantitative milestones to be successful in these application process. We also have a training program for neurotherapeutics development that occurs once a year through an R25 mechanism. And this is the link for the course. But it not only has the ability to go in person for the course annually, all the course lectures are available for anybody in the community. So if you want to learn more about PK or more about tox, you need a little primer on the subject, these videos are available online to anybody in the public to look at. So the last parts I want to do just touch on some things to do and some things not to do. I say this all the time, read the funding opportunity announcement, the NOFO, as carefully as possible. Because every single funding opportunity out there has specific language of things you need to do, and ours is no different. Discuss the program with the program directors, the Home Institute PD program director for the disease interest to make sure you fit in their portfolio, that things go on. If you're going to SBIR, you may want to talk to the SBIR folks as well to compare and contrast Blueprint Neurotherapeutics versus the Omnibus. Okay, stick to the page limits. Don't try to micro font your way through them because you'll get a rejection. you'll get your thing withdrawn, and there's nothing I can do about that when that happens. Make sure you put forth a solid scientific premise with good preliminary data, that it's rigorous, and you need to discuss that rigor in the research strategy section of your application, not bury it in the vertebrate animal section. Obviously, address the criticisms that you can see. Right? If you know it's got a structural alert in the molecule, acknowledge that and talk how you're going to get around that. If you know you have a hole in your disease biology, talk about how you're going to shore that hole up in the first couple of years of the program and what tool combinations you need to make to get there. I strongly suggest contacting a medicinal chemistry expert or BPN staff if you have questions about what those may be. I can give you a general sense of what issues you may face. I can't, again, to avoid IP issues, I can't give you an SAR plan, but I can certainly tell you what I see as general issues that you may need to address. Clearly indicate what will be done as part of the grant and what is expected by BPN contractors. The funding opportunity specifically requests for a table that says that. Here's the assays, here's the work. We're going to do this in our labs, in different co-investigators' labs. We want you, NIH, to do it in these other items ourselves. Include budget sheets for all years of the grants. I get this every round. I get somebody who submits just the UG3 portion of the grant, either in the research strategy or in the budget or both. You have to lay out all 5 years. So that milestone plan I point up is also up on our frequently asked questions on our website that lays out what a 5-year plan should cover from starting point to the clinic. And so it's a good blueprint for moving your stuff forward. Some things to do as well, as I said, explicitly discuss your data, the quality of your data in a detailed manner. Whether it was normalized, whether it was randomized, blinded, what your inclusion exclusion criteria were, was the power analysis done for not only for the experiments that you propose, but also for the experiments that you're citing for preliminary data to help shore that conversation up. If you are coming in new and you've never done this before, start your registration 6 to 8 weeks. So if you're on this call and you haven't done it, today would be a good day to go into the system and start your registration process, because it takes a while to get done. And if you run into glitches, sometimes getting those glitches fixed takes an inordinate amount of time. Sometimes it goes quick, but every once in a while I run into someone that gets stuck in the system, and it could take weeks to get through it. So again, just starting early gives you a chance. If you plan on exceeding 500,000 in direct cost in any year of a UG3 UH3 grant application, you need to seek permission in advance, and 6 to 8 weeks in advance. Again, I'm going to request from you detailed budgets for all 5 years. I'm going to look for what your research strategy is and a budget justification to go with that, so that we can evaluate whether we can get approval to do that. And again, that request may come from NINDS if it's NINDS's mission. It may have to come from NIA if it's NIA's mission, etc. Also important here is the contract costs are not part of your budget. I think I said that before. I'll say it again. Consultants, NIH contracts, not part of your budget. We contract that stuff independently. You don't have to worry about it. That's why that table's there that says, you're going to do this and you ask us to do that, so we can look at the overall impact on what you're requesting, make sure you're not missing anything in your budget that we can't do in ours. Talk with your tech transfer and business development folks. You need a plan for funding those patent and license agreements. Again, don't file your patent too early and create a bunch of prior art. And especially if your folks aren't willing to file for the national filings, that's a big issue that can be addressed if you later in the program, if we have better IP and someone that's with deeper pockets. So again, think about the timing of your patent applications carefully. Things not to do, don't plan for this to be your sole funding for your lab. You know, this program is not designed to fund a lab, it's designed to fund a drug discovery and development program. So since it's milestone driven, it can end abruptly if we run into some sort of, clear no-go decision point. Don't under-resource yourself, as I mentioned, to avoid the budget limits. Be in there 6 to 8 weeks in advance if you need to exceed those budget limits with justification. Do not plan for BPN contracts to do disease biology or one-off in vitro experiments. So if you're coming a development study project and I need you need to go out and get a hERG study, put that as part of your grant to do it, because the amount of time and energy to put a whole contract in place for a single in vitro experiment is prohibitive. So do not code your application. Again, this is really important. Do not code your application as a clinical trial if you plan to use the BPN contractors to do the clinical trial. It's only a clinical trial if you are in fact doing the clinical trial with grant dollars to do that. We will deal with the paperwork and all the stuff that goes with a clinical trial requirements as part of the contracting actions that we do down the road. It also alleviates you from having to give a whole trial design for a healthy human subject clinical trial. You can say in a sentence or two, we're going to do a healthy human subject first in human SAD study followed by MAD study down the road. And that will pretty much cover what you need to really do in most cases. But if you plan to do the trial yourself, it'll be at a minimum a delayed start, and more likely will be a delayed onset clinical trial, because you won't know what you need to do for clinical trial design. And if you're coming in for healthy human subjects, then we should probably have a talk about why you want to do it yourself through your contracts versus just using ours. So that's the last slide and things I have to do. I did promise I would cover BPN biologics. The major difference biologics, obviously it covers different modalities like antibodies and peptides, proteins, gene-based therapies, cell therapies. It has the same receipt date as BPN, August 9th. It has different PAR numbers associated with it, but it has both the UG3 and UH3. The biggest difference is, because of the nature of biologics, a 2-year UG3 is allowed, unlike small molecules, in order to get to sort of that same data package to make the decision to move forward. Chris Boshoff is the contact for this, and like the BPN, it has its own web page. In fact, if you come to the small molecules, you can click over to the biologics from that same page, as well as for devices. There's a MedTech program there too. Lastly, if you're in the pain therapeutics development area, we have a sister program that is funded by the Helping to End Addiction Long-Term HEAL initiative from NIH. Marianne Palamater runs that program. It has receipt dates 3 times a year, so the next one is June. And it is set up identically, with the exception of 2 things. You can have both biologics and small molecules under the same FOA, and you can do more stuff yourself here, but it has a requirement for a pharmacodynamic target engagement biomarker as part of your planning, and how you might be able to address that. It could be funded through the program. So again, if you're interested in that in pain therapeutics area, you can do Marianne Palamater. It takes not only stuff from all the neurosciences, it takes any pain indication anywhere in NIH. So I apologize. I just wanted to noise I was getting. And so the end goals is all the same: get to clinical trial, hand it off, academics, small businesses, all there. So at this point, we'll open it up for discussions. And I could talk about the application process, the review process, proposals, contracts, budgets, intellectual property, any and all of that. And lastly, if you need to contact me, here's my email. So I'll take questions now.
Thanks, Chuck. So before I read the first question, let me remind everyone that this was recorded and it will be sent out along with the presentation slides in the next upcoming days. And also, please submit your questions in the chat box. So the first question is, what do you mean by benchmarking your compound?
Sure. Benchmarking a compound. So a lot of times people come in and say, I have the starting point. And all they'll know is it is, active in this in vitro assay, or they will know that it has activity in a in vivo assay. So benchmarking the compound is looking more broadly. So looking at, is it active in the vitro if I only have in vivo and vice versa? It is looking at in vitro metabolism, maybe we're running um, rodent PK. It's things like off-target screening. It could be doing some early preliminary SAR to see whether I can show signs of SAR if I've only got a hit compound, that it's not completely uh, biphasic, meaning that I either have activity, I have no activity, and I can't find anything in between. So really getting a better feel for that compound and that assays, that we have a good set of benchmarks on that. It's also from the assay point of view, benchmarking the assay, making sure it's performing as needed for optimization efforts. So if it's a 96 well plate format assay, is the C prime looking good for that? Um, and so is the assay itself performing well?
Okay. Next question is there a set of criteria and milestones more specific to ASOs?
Um, I don't think so. I mean, the ASOs will be probably most appropriate in the biologics side of the BPM biologics. Um, eh, but the criteria is going to be similar.
Okay, I'm getting a few questions about what is the last application date for the current funding opportunities and what if they need to resubmit?
Okay. So as you saw from BPM Biologics, it's a later PAR, and so that will have another receipt date in February 9th of 2024 already announced. What I can say about BPN small molecules is that the last receipt date on the present PAR is August 9th. I just did concept clearance for renewal of the funding opportunities publicly at the May Council of NINDS. So it's out in the public that our intent is to go ahead and reissue those. But I haven't put out a public notice yet to that effect, which will come next.
Although a project may not be a clinical trial using the BPN contractors, would it still be considered human subjects research for the purposes of NIH and NIH human subjects form submission?
If you're doing it through our contractors, the clinical trial piece, it is not human subject or human clinical or clinical trial. Those forms you're discussing will be part of the contract work that is done. It is still possible to have human subject work done that's not related to clinical trial. For instance, I had someone that was collecting human uh, blood and using that for an assay. Um, so they did, in fact, have human subjects that they were collecting blood from to create their in vitro assay, and they had to have the forms associated with doing it for that. So it is still possible to have human subjects up, but if you're talking strictly about the clinical trial, no.
Okay, will BPM biologics allow combinations of biologics and biologics that are being developed as a biologic device combination?
I don't want to speak too much for the biologics programs. I would refer you to Chris Oshoff to do this. Combination projects, products are inherently more difficult. And the answer to that question is not a simple one. You know, is one already approved? Are both already approved? Will or both new? Is the device approved or not approved? All dictates what's required for to get to that first in human trial. So if it starts to be, multiple toxicology studies, multiple entities, um, normally that we would not do that kind of project as a combined project. We would sort of take it as one bite at a time.
Okay, next question. Are exosomes part of this program or biologics?
Would not be would not be small molecule. And again, I would refer you, before you came to BPM Biologics, I would refer you to David Jett in our Division of Translational Research, who is our expert in that area, and have a discussion with him about your project and where he thinks the best fit for your program is.
I'll put David Jett's contact information in the chat momentarily. So if I already have a company interested in a clinical trial in Italy and I have a R61, R33 in review, should I consider the BPN? Also, my compounds are already FDA approved.
If you're already ready for the clinic and they're already FDA approved, no, I would not consider the BPN. Because anything directed beyond phase 1 and anything that's just for clinical trials out of scope for the BPN. There are other funding mechanisms that you could apply depending on the indication area. NINDS has exploratory clinical trial funding opportunity that you could look into. There's also the SBIR program.
Carolyn, you can go. I don't have any more right now.
Yeah, okay. Does BPN also have a program to support phase 2 clinical trials?
It does not. As I mentioned, there are in the Division of Clinical Research at NINDS, and again, it depends on which IC you're doing. Multiple ICs have programs that support clinical trials in the phase 2 efficacy study area. That includes the exploratory clinical trial program, as well as multiple networks in NINDS for different indications.
Okay, if we already have selected 2 compounds based on in vivo efficacy, what's the appropriate point of entry? If I enter in discovery, can I optimize the assay?
Not sure what you mean by optimized assay if you pick 2 compounds, but I'm going to guess what you're trying to say is, and you can always clarify it, is I have 2 compounds and trying to figure out which one to pick of these 2. Can I do?
So I can add more, Chuck. If they optimize the assay in preparatory phase and then select lead compound with the assay for an in vitro admit in the discovery phase.
Yeah, I mean, it progresses. Like I said, I would look carefully at our milestone progression. It's standard fare to come in there, but if you're down to your final 2 compounds, that's often just done in the preparatory phase. We'll discuss how to, do what it's going to take to differentiate between these 2 compounds and what assays need to get run to do that. So again, like having a call with NIH staff, myself, or others would be useful to really get into more of the details of that particular question, because it's not a simple one.
If they just want to do up to IND filing, and can they do that with BPN and then do clinical trial later with other funds?
It's not the general goal of our program to do that. Our general program goal of the program is to take it into the clinic. It's theoretically possible that in cases we have stopped at IND filing, but you can't file an IND without a clinical site. Because part of the IND filing is Form 1571 and 1572 from your clinical site.
Okay, if we have the amino acid sequence, can we ask BPN contractors to make Thai compound for us?
Make what compound?
Thai, TYE? Or maybe they meant to make the compound for us.
Okay. If you have the amino acid sequence and you just wanted them to scale this up, so I'm assuming I'm assuming that's a development compound. You're asking an outside contractor to scale it. It could be done through, depending on the size of the peptide, it would either be in BPN biologics or BPN small molecules. So if you're less than 6 amino acids, you would contact BPN. If it's more than 6 amino acids, we would contact BPN biologics program. And depending on what you're actually looking for, it may or may not be more appropriate for the biologic side, which has different contracts.
What do we need to contact the PO now? Draft of the proposal, or do you have a template for the letter of interest?
I don't have a template for the letter of interest. I do have a standard form I give folks to fill out for our pre-call. Raconda schedules all my calls, so requests come in and Raconda gets them on my calendar. So I usually ask for two things: a set of specific aims, so I understand where it is you are and where you want to go, and some background information that's on this Excel form that we give you to fill out, and then we can have a call. Both are about 1 page, so we're talking like 2 pages of information.
Okay. There was another one about that. So if I think they're getting at, can the IC talk to me and offer guidance on entry stage? What information should I provide before the call? So
Yeah, yes, we can, and often it's what a lot of what our conversations are in the pre-calls are trying to determine where the best entry point is. As I said, the milestone layout, along with sort of the resources that are available and all that, are on the web page, and I strongly recommend you look at that web page. But it would be so. Let's see if I can move this over here and still see it. Don't. Okay. So this is that web page on here. If you go down to the frequently asked questions page and you go down to the sort of the bottom, how do I develop my milestones for my project? You will see there's a bunch of information, but there's this click right here on the BPN milestone things, and it has what was in their slides, but it has a lot more detail in there. It talks about what our standard milestones look like. It also talks about what resources are available BPN at each stage. And we'll give you sort of what's typical for us to provide and what's typical for you to see for milestones across. Obviously, your milestones may be modified from these to have your specific project in mind, but you can walk all the way from the beginning to the end and what resources are potentially available to you from BPN. It'll also help you figure out where your entry point is, because you can determine, do I have this or not? Well, I'll put you someplace not. As I said early on, it's better not to try to say you're too far down the pipeline when you're not, because we'll put these milestones. If you can't meet them in the timeline, you end up getting stopped. And we can do some adjustments to timelines, but not add more money. We have the ability in the time, quality, and money situation, right? We control the money, but we can't really add a lot more to it. Very limited ability to do that. Time, is the one component. We can sometimes give no cost extensions to meet a milestone if in fact things are progressing and there's money still available. But we're not going to obviously compromise on the quality.
Okay. Is BPN appropriate for repurposing of a compound that failed a clinical trial and is being tested for a different indication?
I would need to know more details. Repurposing of a compound is often quite difficult on many fronts. Do you own intellectual property? Is there any ability to develop intellectual property? Can I actually find a commercialization for that repurposed drug at the end of the road? Because again, if there's no intellectual property and no ability for them to recoup their money, it's hard to get investors to invest in repurposing of a drug. If it's a marketed drug repurposed for something, then it's typically something that's could go directly to phase 2, and I'd encourage you looking at mechanisms from the Division of Clinical Research to actually test that hypothesis out. So it's a difficult road to repurpose a compound. You should talk to myself or other program officers about the path you're proposing and whether we could see some way to do that.
We're getting really close to time, so I'm going to ask a few more, but there's a lot of questions about Ignite, the R61, R33, and I will reach out after and connect you with the relevant program staff.
Yeah, if you could put Becky Roof's contact information up on the chat so people have it.
Yep.
And eventually the slides will come out. You'll see her contact information there as well.
One more. Are BPN consultants and CROs available to talk about an application plan?
We don't pay them to talk about to prospective grantees. It's the best way I could put that. I can't stop you from talking to anybody you wish to talk to, but they won't talk to you for free in most cases.
I think we have one more. Do you need to have a backup compound if you're entering at lead optimization? Also, if optimization is specifically related to drug-like properties and not efficacy, for example, compounds were phenotypically screened and mechanism of action is unknown, is this a problem for the program?
No, it's not. It is for one reason, for a couple of reasons. We don't pay for backups to our original thing. So if you have a backup program, we usually say bring that in through a different application process. We oftentimes develop more than one series, and we pick the series that is the best one to move forward, and the other one gets shelved unless we run into an issue in the lead optimization phase to get towards development candidate. It is okay to have phenotypic. You're going to have to make a case for the efficacy, the animal efficacy study, or whatever the reason why you believe it's going to be working in humans down the road. It is okay to do optimization as long as you have the assays, to drive that optimization in place. Either we can certainly do the PK optimization through in vitro ADME as well as in vivo ADME work. But you would need to have the disease biology assays in place to test them as we make those changes. Efficacy is not going off to. So it becomes a more expensive in many cases, but it is definitely doable.
Okay, thanks, Chuck.
All right. Again, reach out via email. If you have Rakonda's email, can CC Rakonda on the outreach, and she'll know. If not, I'll forward it to Rakonda as I get them and see them to get them set up for scheduling on my phone screening. Good luck, and I really appreciate everyone taking the time today to listen to the seminar and learn more about our program. We're here to help. Bye now.
Thanks, everyone. Have a good one.